CSIR Evolution Chapter 6. Chapter 6, Molecular Evolution. Biologists are developing a comprehensive picture of the evolution of life on Earth through the combined knowledge obtained from molecular data and fossil records. Molecular evolution studies evolutionary change at the level of the DNA sequence. Molecular evolution falls into three areas. First area is determining the extent and causes of genetic variation in natural populations. Molecular techniques allow these matters to be addressed directly by examining sequence variation in proteins and DNA. A second area of research examines molecular processes that influence evolutionary events, and the results of these studies have elucidated new mechanisms and processes of evolution that were not suspected before the application of molecular techniques to evolutionary biology. The fate of those mutations that affect the fitness of their carrier is partly determined by natural selection. On one hand, new alleles that confer a higher fitness tend to increase in frequency over time until they reach fixation, thus replacing the ancestral allele in the population. This evolutionary process is called positive or directional selection. Conversely, new mutations that decrease the carrier's fitness tend to disappear from populations through a process known as negative or purifying selection. Finally, it may happen that a mutation is advantageous only in heterozygotes but not in homozygotes. Such alleles tend to be maintained at an intermediate frequency in populations by way of the process known as balancing selection. For an organism, a mutation, besides exerting a positive or negative effect, or its effect can also be neutral, in the sense that could have no adaptive value. In this case, natural selection would not work on it, and its destiny would be determined by the only other existing mechanism, i.e., by genetic drift. It is necessary to estimate that how many neutral mutations occur in nature, on average, in respect to adaptive ones, in order to understand evolution at molecular level. Indirect observations. An estimation of the ratio between neutral and adaptive mutations are logically needed to be estimated, since direct measurements were impossible, and the ratios must be fairly close to the ratio between neutral and adaptive phenotypic characters. A third area of research in molecular evolution applies molecular techniques to constructing phylogenies of various groups of organisms. A detailed evolutionary history is found in the DNA sequences of every organism, and molecular techniques allow this history to be read. A typical example, in this field, is the comparison between amphibians and mammals. Both the amphibians and mammals are derived from a common evolutionary ancestor, but the evolution of amphibians was much slower than that of mammals. These life forms share so many anatomical characters that a single order comprises them all, while mammals differentiated in as many as 16 distinct orders. Mammals clearly underwent a much faster phenotypic evolution than amphibians. It is logically possible that mammals have undergone much faster genetic changes than that by amphibians. From the above and many other similar observations, it was concluded that natural selection is the collaborative result of phenotypic evolution, molecular evolution, and macroevolution, where genetic drift plays a minimal role. This theoretical conclusion, known as selectionism, or more precisely, panselectionism, became increasingly popular in the 1940s and 50s. As molecular research progressed, it was observed that the results obtained from selectionism were quite different from the actual results when the direct methods for measurements of molecular parameters. The measurements made at molecular level suggest that mutations accumulate in the genomes of amphibians and mammals at the same rate. The rate of molecular evolution was the same for both species, but the rate of phenotypic evolution was quite different. It is not worth correlating the phenotypic changes with molecular changes, and thus, both of the levels must be kept apart. This creates, of course, an additional problem. Because now we have to find out a link between these two hypotheses. 6.1 Use of molecular data for evolution. Evolutionary genres was confined largely to the study of how phenotypes change with the passage of time, as it was not possible to examine genes directly for many years. Investigation of evolutionary changes is now possible by directly analyzing protein and nucleic acid sequences as the molecular genetics branch has been tremendously advanced. These molecular data offer a number of advantages for studying the process and pattern of evolution. 1. Heritable, genetic changes accumulate over time and eventually result into evolution. Although the relation between genes that have undergone changes and the trait may be complex, anatomical, behavioral, and physiological traits often have a genetic basis. Changes in the protein or nucleic acids directly have associated genetic changes. 2. Present in all organisms, although early studies of population genetics relied on simple genetic traits such as human blood types or banding patterns in snails, which are restricted to a small group of organisms, but genetic data can be obtained for all living organisms as all of them have proteins and nucleic acids. 3. High polymorphism, a large amount of data can be accessed by molecular methods. For example, the human genome, which contains more than 3 billion base pairs of DNA, constitutes a large pool of information about our evolution. 4. Allows comparison of distinct organisms, since distantly related organisms have a very few characteristics in common. Thus, trying to assess their evolutionary history or relatedness is often difficult. Comparison of floral anatomy was the traditional way to find out the evolutionary relationships between angiosperms, whereas the evolutionary relationships of bacteria were determined by their nutritional requirements and staining properties. Because plants and bacteria have so few structural characteristics in common, evaluating how they are related to one another was difficult in the past. Certain molecular traits such as ribosomal RNA sequences and some fundamental proteins are common in all organisms. Such molecules are conserved during evolution and they provide valid basis for comparisons among all organisms. 5. Quantifiable, protein and nucleic acid sequence data are precise, accurate, and easy to quantify, which facilitates the objective assessment of evolutionary relationships. 6. Provide information about the process of evolution. The process of evolution can be better studied on the basis of molecular data. For example, in mosquitoes, the study of DNA sequences reveals that insecticide resistance has probably been arose from a single mutation, which subsequently got spread throughout the world. 7. The database of molecular information is large and growing. Today, this database of DNA and protein sequences can be used for making evolutionary comparisons and inferring mechanisms of evolution. 6.2 Protein variation, evolution, and animal behavior. To study the process of evolution, study what type of genetic variation has occurred and up to what extent it has affected the natural population are very crucial. In case of some traits, the interaction of many genes and various environmental factors have role in determining the phenotype. Initially, population genetics was forced to rely on the phenotypic traits that had a simple genetic basis, such as human blood types or spotting patterns in butterflies, Figure 47. Figure 47, variation in the spotting patterns of the butterfly Panaxia diminula. The first clue about direct examination of molecular evolution came from the application of electrophoresis to population studies. Using electrophoresis, macromolecules, such as proteins or nucleic acids, can be separated on the basis of their size and charge. In 1966, Richard Lewontin and John Hubby obtained proteins from wild fruit flies, separated them by electrophoresis and stained for specific enzymes, and observed the separated pattern of bands on gel. By examining the patterns of bands on the gels, they assigned genotypes to individual flies and quantified the amount of genetic variation in natural populations. Harry Harris quantified genetic variation in human populations by using the same technique in the same year. By using the same technique, hundreds of different proteins variants have now been examined. Figure 48. Figure 48, molecular variation in proteins is revealed by electrophoresis. Tissue samples from Drosophila pseudoobscura were subjected to electrophoresis and stained for esterase. Esterase encoded by different alleles migrate different distances. Shown on the gel are homozygotes for 3 different alleles. Measures of genetic variation. 2 parameters are used to measure the amount of genetic variation in populations. First is the proportion of polymorphic loci, which is the proportion of examined loci in which more than one allele is present in a population. For instance, if 30 different loci were examined and 2 or more alleles were found at 15 of these loci, then the percentage of polymorphic loci would be 15 divided by 30 is equal to 0.5. Second parameter is the expected heterozygosity, which is the proportion of individuals that are expected to be heterozygous at a locus under the Hardy-Weinberg conditions, which is 2pq when there are 2 alleles present in the population. In case of observed heterozygosity, breeding system of an organism plays a role and thus varies from the expected heterozygosity. Therefore, the expected heterozygosity is considered in calculations. For example, there is little or no heterozygosity in case of self-fertilizing species. In such cases, some considerable genetic variations are still observed, which are detected by the expected heterozygosity. Typically for a number of loci, expected heterozygosity is calculated and is then the average heterozygosity is calculated for all the considered loci. For a number of species, the expected heterozygosity and percentage of polymorphic loci have been determined by protein electrophoresis. About 1/3 of all protein loci are polymorphic, and expected heterozygosity averages about 10%. Although there is considerable diversity among species, the true amount of genetic variations are actually underestimated due to these measures because protein electrophoresis does not detect some amino acid substitutions, nor does it detect genetic variation in DNA that does not alter the amino acids of a protein. Reason for protein variation, although the evolutionary significance of large amount of genetic variations was not at all clear, geneticists found large amounts of genetic variations in most populations to account the presence of extensive molecular variations in proteins. 2 mutually opposing hypotheses arose. 1. Neutral mutation hypothesis. Electrophoresis reveals high incidence of polymorphism, which may not be important from an evolutionary point of view. If all or most electrophoretic alleles are neutral, i.e., if no allele is more fit than its alternative, or only very slightly deleterious, there is virtually no selection at these loci, and the variation observed in the population is merely a chance accumulation of a combination of mutation and genetic drift. Kimura, a Japanese scientist, proposed this model, which is an alternative to the natural selection model, in 1968. It proposed that the molecular variation revealed by protein electrophoresis is adaptively neutral, that is, individuals with different molecular variants have equal fitness. This hypothesis assumes that all the variants of a protein obtained in electrophoresis are functionally equivalent and none of them is functionless. Because these variants are functionally equivalent, natural selection does not differentiate between them, and their evolution is shaped largely by the random processes of genetic drift and mutation. The neutral mutation hypothesis accepts that natural selection is an important force in evolution, but views selection as a process that favors the best allele while eliminating others. It proposes that, when selection is important, there will be little genetic variation. According to neutral mutation hypothesis, 1. The rate of evolution for most genes will be equal to the neutral mutation rate. 2. Advantageous mutations are exceedingly rare. 3.at The molecular level genetic drift dominates evolution. 2. The balance hypothesis. According to this hypothesis, the genetic variations in natural populations are maintained by selection that favors variation, balancing selection. An example of balancing selection is given by overdominance of heterozygote in a population. In this type of population, a heterozygote has higher fitness than that of either homozygote, is one type of balancing selection. According to this hypothesis, molecular variants are not physiologically equivalent and do not have the same fitness. Instead, genetic variation within natural populations is shaped largely by selection, and, when selection is important, there will be much variation. 6.3 Pattern of molecular variations. The field of detection, quantification, and investigation of genetic variations has progressed rapidly over the past few years. It has added much information and deepened our knowledge and understanding of the diversity of genetic variations in the different genome. The application of many techniques has provided a detailed view of molecular variations. To detect genetic variations in DNA and examine patterns of genetic variations in nature, restriction enzymes are one tool that can be used to detect genetic variation in DNA and examine patterns of genetic variation in nature. Each restriction enzyme recognizes and cuts a particular sequence of DNA nucleotides, known as that enzyme's restriction site. The method, which involves use of restriction enzymes to recognize the variation present in restriction site, is called as restriction fragment length polymorphism, RFLP. A restriction enzyme may act at a few sites within the DNA. But, in RFLP, a number of different restriction enzymes can be used in combination to estimate the amount of variations in the DNA and the proportion of nucleotides that differ between organisms. Amplified fragment length polymorphism, AFLP, variable number of tandem repeats, VNTR, and single nucleotide polymorphism, SNP, are some other molecular markers used for analysis of DNA variation. The significant progress has been driven mainly by the developments of microarray and next generation sequencing. The molecular studies of numerous genes suggest that the rates of evolution of different genes or different parts of the same gene are often variable. Rates of evolutionary change in nucleotide sequences are usually measured as the rate of nucleotide substitution, which is the number of substitutions taking place per nucleotide site per year. To calculate the rate of nucleotide substitution, we begin by looking at homologous sequences from different organisms. One can compare and find out the differentiating nucleotides between two or more homologous sequences. For example, the growth hormone sequences for mice and rats can be compared, which diverged from a common ancestor from about 15 million years ago. Nucleotide changes in a gene that alter the amino acid sequence of a protein are referred to as non-synonymous substitutions. If the nucleotide substitution is at the third position of the codon, they usually do not alter the amino acid sequence, and such mutations are called synonymous substitutions. The rate of non-synonymous substitution varies widely among mammalian genes. The rate of non-synonymous substitution for the actin protein is only 0.01 by 10 substitutions per site per year, whereas the rate for interferon Y is 2.79 times 10, about 1,000 times as high. The variation in the rate of synonymous substitutions among genes is far less. For most protein-encoding genes, the synonymous rate of change is considerably higher than the non-synonymous rate because synonymous mutations are tolerated by natural selection, Table 7. Non-synonymous mutations usually are not tolerated because they alter the amino acid sequence of the protein. Such altered proteins in many cases have a negative effect on the fitness of an organism, therefore natural selection tends to eliminate the most of these mutations. Table 7, Rates of non-synonymous and synonymous substitutions in mammalian genes based on human rodents. Comparisons. The rate of evolution in protein-coding genes is commonly assessed with the two quantities dn, rate of non-synonymous substitutions per non-synonymous site, also called co, and ds, rate of synonymous substitutions per synonymous site, also called k2. The ratio of the number of non-synonymous to synonymous substitutions per site, dn/ds, identifies the type of selection pressure acting on a gene. dn/ds is less than 1 indicates strong purifying selection or stabilizing selection acting against change. dn/ds is more than 1 indicates positive or Darwinian selection, driving change. dn/ds is equal to 1 implies that amino acids evolve largely neutrally, no change. Not only two different genes, but different parts of a single gene also evolve at different rates, with the highest rates of substitutions in regions of the gene that have the least effect on function, such as the third position of a codon, flanking regions, and introns, figure 49. Though when a matured mRNA is formed, the 5' and 3' flanking regions of genes are not transcribed into RNA, and therefore substitutions in these regions do not alter the amino acid sequence of the protein, although they may affect gene expression. Although introns do not encode for any amino acids, they have a higher substitution rate than that for exons. Introns are flanked by particular sequences at the 5' splice site, 3' splice site, and have branch point within their sequence, which are required for correct splicing of the pre-mRNA to form a matured RNA which encode a functional protein. Figure 49, the highest rates of nucleotide substitution are in sequences that have the least effect on protein function. Introns, while least substitution are in sequence, which have the maximum effect on the protein function, exons. The 5' untranslated region and 3 untranslated regions, 5 UTR and 3 UTR, of a gene have low substitution rates relative to other sequences. These untranslated regions, though appear in the mRNA, but do not get translated. Instead, the 5' untranslated region contains the ribosome binding site, which is essential for translation, and the 3' untranslated region contains sequences that may function in regulating mRNA stability and translation. Therefore, substitutions in these regions are usually not tolerated as they have deleterious effects on fitness of an organism. Non-synonymous substitutions are observed at their lowest rates in the coding region because these substitutions always alter the amino acid sequence of the protein, which often has deleterious effects. On the other hand, pseudogenes, which are duplicated non-functional copies of genes that have acquired mutations, have highest rates of substitution. These altered pseudogenes can no longer produce a functional product, therefore mutations in pseudogenes do not affect the longevity of cell's life. In short, it can be said that function of a gene and its rate of evolution are correlated, such that sequences which are not functional are prone to substitution than the functional or geneic sequences. This observation fits with the neutral mutation hypothesis, which predicts that molecular variation is not affected by natural selection. 9.4. The molecular clock. The neutral mutation theory proposes that evolutionary change at the molecular level occurs mainly by the fixation of neutral mutations by genetic drift. The mutation rate should be fairly constant for any particular gene, and it provides the basis for replacement of neutral mutation by another neutral mutation. The amount of molecular change a protein has undergone can be used as a molecular clock to mark evolutionary events when the rate of evolution of a protein remains roughly constant over time. For example, the enzyme cytochrome C tends to be conserved throughout evolution, and hence it can be used to examine two organisms which are claimed to be derived from a common ancestor from the fossil records. Comparison of the number of differences in the cytochrome C amino acid sequences in each organism can help one to estimate the number of substitutions that have occurred per amino acid site. If the fossil records state that the two organisms are diverged about 400 million years before, and molecular analysis finds that there are 20 amino acid substitutions, then it indicates that an average rate of 5 substitutions per 100 million years. Knowing how fast the molecular clock ticks allows us to use molecular changes in cytochrome C to date other evolutionary events. For an instance, if one finds that two organisms have cytochrome C molecules that differ from each other by 15 amino acid substitutions, then our molecular clock would suggest that they diverged some 300 million years ago. If we assumed some error in our estimate of the rate of amino acid substitution, statistical analysis would show that the true divergence time might range from 160 million to 440 million years. The molecular clock is analogous to geological dating based on the radioactive decay of elements. Emil Zuckerandi and Linus Pauling in 1965 proposed the molecular clock in order to date evolutionary events on the basis of molecules in present-day organisms. A number of case studies have examined the rate of evolutionary change in proteins, and the molecular clock has been widely used to date evolutionary events when the fossil record of absent or ambiguous. Figure 50 represents relation between the rate of amino acid substitution and time since divergence, based in part on amino acid sequences of alpha hemoglobin from the 8 species shown in Figure 50b. The constant rate of evolution in protein and DNA sequences has been used as a molecular clock to date past evolutionary events. Phylogeny of 8 of the species that are plotted in Figure 50a and their approximate times of divergence based on the fossil record are represented in phylogenetic tree shown in Figure 50b. Figure 50. The molecular clock is based on the assumption of a constant rate of change in protein or DNA sequence. But several molecular studies reveal that the ticking of molecular clock is not always at a constant rate, particularly over shorter time periods, and this method remains controversial. When the amino acid sequences are known, they can be used to estimate the rate of evolutionary V change. That is, the data show how many amino acid substitutions have occurred between two known groups of organisms. From the knowledge of genetic code, the minimum number of nucleotide substitutions required for a change can be estimated. For example, if the seventh amino acid in a protein is phenylalanine, codons UUU, UUC, whereas the same protein in different species has an isoleucine at the same position, AUU, AUC, AUA, then the minimum number of substitutions to convert a phenylalanine codon to an isoleucine codon is 1, UUU, AUU. We can calculate the evolutionary rate, nucleotide substitutions per million years, when we know the minimum number of substitutions. Such observations enable us to build a molecular evolutionary clock which evolutionary time in nucleotide substitutions can measure. There are various proteins such as hemoglobin, cytochrome C, blood clotting factor fibrinopeptides, etc., which are extensively studied in terms of molecular studies in terms of nucleotide substitutions and hence evolutionary rate. The way how does the amino acid sequence differ among species is shown in Figure 51. From such comparisons one can calculate the actual number of amino acid differences, as well as percentage differences. Figure 51, the amino acids making up the terminal portion of cytochrome C in 3 species. The combined representation of percentage differences of cytochrome C of various species is shown in Table 8. This type of information can be used 2 ways. First of all, the phylogenetic tree of species under consideration is constructed, which can explain about the evolutionary history of the species, Figure 52. Then this tree can be compared with the trees obtained from other means such as those obtained from fossil evidences or evidences from morphology, physiology, and development. Such comparisons can give an idea about the areas of disagreement in an attempt to find out the best way to create phylogenetic trees. Molecular Phylogeny can also give anyway unattainable information about the organism even when the fossil records are incomplete or improper. Figure 8, amino acid difference, percentage, in cytochrome C between different organisms. The average rate of substitution can also be calculated from DNA or amino acid difference data if the current amino acid differences in the proteins of two species are known. The actual number of nucleotide substitutions that have taken place over evolutionary time can be appropriately estimated by using the statistical Poisson distribution, which deals with rare events. The average number of amino acid substitutions between two proteins per site are represented by the index K. K equals ln 1p, where n is the natural logarithm to the base e and p equals d/n, in which d is the number of amino acid differences and n is the total number of amino acid sites being compared. For example, in Figure 6, n equals 8 and d equals 3 between the dog and chicken. Thus, 0.375 equals 0.47. K equals ln 1. The average number of amino acid substitutions per site between dog and chicken is thus found to be 0.47. Figure 52, composite evolution of hemoglobin, cytochrome C and fibropeptide A. The total number of nucleotide substitution appear on horizontal axis. For example, if two species are estimated to be evolved from a common ancestor at about 9 million years ago, and if the rate of divergence per base pair is estimated to be 0.0015 per million years, the proportion of base pairs that differ between the two species at present can be calculated. Here, the rate of divergence per base pair is given to be 0.0015 per million years, so in 9 million year changes in each species will be 9 times 0.0015 is equal to 0.0135. Total divergence among species A and B will be 0.0135 plus 0.0135 is equal to 0.0270. One further step towards the calculation is determining rate per year. K minus K divided by 2T, where K is the amino acid substitution rate per site per year, and T is the number of years since the two species diverged from a common ancestor. The term 2T suggests that each side of the tree has evolved independently for T years. When Ks are calculated for many proteins over many species, they cluster around 109. In honor to Linus Pauling, who first proposed the concept of molecular clock along with E. Zuckerkandl in 1963, Kimura has suggested the unit of a Pauling to be equal to 10 amino acid substitutions per year per site. 109 would be the rate of the molecular evolutionary clock if the values of K form a normal distribution around 10. So far, the data have been too limited to determine the distribution. Despite a few controversies, neutralists claim that molecular evolutionary clock is considered to be relatively constant and as a strong evidence in support of the neutral gene hypothesis. If the rate of molecular evolution is constant over many different time intervals for many groups of organisms, then it reflects that the substitution rate is not a directed or selectional process, instead it is an opportunistic and random event. This doesn't mean that no adapted changes occur in proteins or that there are no constraints. In fact, the evidence suggests that three classes of amino acids can be grouped in terms of substitution rate: invariant, moderately variant, and hypervariant. It's more logical that amino acids with an active site of the enzyme are not virtually substituted since any amino acid change, because amino acid substitutions in that area are not tolerated as they are deleterious or lethal. For example, cytochrome C proteins studied in case of all organisms show that the segment from amino acid 70 to amino acid 80 is necessarily invariant. It is seen conserved in all organisms as it forms the binding site of the protein. 6.5 Evolution of genomes. The entire set of all genes present in an organism's body is called as its genome. Generally, most of genes are present on chromosomes within nucleus, but there are some extra chromosomal genes present on plastid and mitochondrial DNA. In case of sexual reproduction, mitochondrial or chloroplast genes are transferred from female germ cell, but nuclear genes are transferred to the next generation from both the parents. Sexual reproduction results into shuffling and remixing of genes due to meiosis and fertilization in every generation, and individual gametes are transmitted as partly randomized subsets of parental genes to offspring. For establishment of successful shuffling, genes must be able to operate in a wide variety of genetic backgrounds. Usually closely linked genes, or genes whose functions are related to some particular character, are grouped together. For instance, genes involved in certain developmental pathway are closely linked on chromosomes and are inherited together. A gene will not be passed on to successive generations unless the individual with the gene survives and reproduces. Therefore, the gene's probability of transmission is increased by its capacity to cooperate with different combinations of other genes. For this reason, it is useful to view the genes of an individual as interacting members of a group, among which there are divisions of labor, but also strong interdependencies. Genomic evolution deals with questions like, how do proteins acquire new functions? Why are the genomes of different organisms so variable in size? How has enlargement of genomes been accomplished? Because they look at the genome of an organism as an integrated whole, data about the organism. 6.5.1 The evolution of macromolecules. While studying molecular evolution, one must study the evolution of macromolecules, while the outputs of the study are used to reconstruct the evolutionary history of genes and the organisms that carry them. The biomolecules, which are frequently used by molecular evolutionists, are nucleotides, nucleic acids, amino acids, and proteins. Nucleic acids evolve by means of nucleotide-base substitutions, which in turn can result in changes in the amino acids they encode. Changes in the ordering of amino acids, which constitute the proteins, result in the changes in the protein structure. Molecular evolutionists characterize the precise structures of these macromolecules, nucleotide sequences in nucleic acids, and primary structure in proteins. The information obtained from studies is used to determine why and how the macromolecules have been changed. The first step in recognizing the causes of molecular changes is obtaining the phylogenetic information. On the other hand, knowing the pattern and rate of change of a given macromolecule is very important in order to reconstruct the evolutionary history of groups of organisms. 6.5.2 Proteins acquire new functions. Evolution as we know it would not have been possible if proteins were unable to change their functional roles. The earliest forms of life must have had very few genes and proteins. Because much evidence indicates that all living organisms arose from a single ancestral lineage, the many thousands of different functional genes in modern organisms must have arisen from those few ancestral genes. 1. Proteins may acquire new functions via gene duplication. To acquire new functions, gene duplication have important role. After duplication, a copy of gene is freed from performing its original function. Instead, it becomes redundant if the original gene is still producing the original protein. Therefore, entirely novel proteins can be produced without affect the normal physiology of the cell by gene duplication, which may involve part of a gene, a single gene, a portion of a chromosome, an entire chromosome, or the whole genome. The process of speciation is greatly dependent on duplication of the entire genome, polyploidy, especially in case of plants. Since all of the chromosomes are duplicated, generally the autopolyploid individuals are viable because due to duplication of entire set of genome, imbalances in gene expression are reduced. Besides gene duplication, which we are going to discuss, there are some other ways through which proteins can acquire new functions. 2. Physiological changes may lead to the evolution of new functions for a protein. Lysozymes are the enzymes which digest the cell walls of bacteria and rupture their plasma membranes, thereby killing the cells. These are the hydrolytic enzymes found in almost all animals. Lysozymes are produced in the tears, saliva, and milk of mammals and in the whites of bird eggs. Due to their degradative activities, lysozymes play an important role as a first line of defense against invading bacteria. Since all organisms have developed mechanisms to eliminate invading bacteria, therefore probably most of the animals have lysozymes. The digestive properties of lysozymes are also useful for some animals in the digestion of their ingested food. In some mammals, a mechanism of digestion is developed twice and is called as foregut fermentation. Mostly foregut fermentation evolved independently in ruminants, a group of hoofed mammals that includes cows and other cattle, and in certain leaf-eating monkeys, such as langurs. Here, the foregut or the posterior esophagus and/or the stomach have been converted into a chamber in which the gut microflora bacteria break down ingested plant matter by fermentation. Foregut fermenters can obtain nutrients from the otherwise indigestible cellulose that makes up a large proportion of the plant body. Since evidences suggest that the close relatives of langurs and ruminants have not evolved with foregut fermenters, one can conclude that these evolutionary events were independent. In both foregut fermenting lineages, the enzyme lysozyme has been modified to play a new, non-defensive role. Lysozyme ruptures some of the bacteria that live in the foregut, releasing nutrients that the mammal absorbs. Lysozymes are non-functional when produced but become activated after a sequence of changing events, such as exposure to acidic conditions of the mammalian foregut. To answer this question, molecular evolutionists compared the amino acid sequences of lysozyme in foregut fermenters and in several of their non-fermenting relatives and could find several amino acids differing. Table 9. Shown above the diagonal line is the number of amino acid sequence difference between the two species being compared. Below line are the numbers of sequence uniquely shared by the two species. Asterisk indicates foregut fermenting species. The studies also involved comparison of the pattern of these changes with the phylogenetic relationships among the species that had been established based on fossils and current morphology. An important outcome of the study was the finding that without the occurrence of gene duplication, lysozymes had some amino acid changes in lysozyme, which occurred in the absence of gene duplication, happened about twice as rapidly in the lineage leading to langurs as in any other primate lineage. The studies revealed that lysozyme went through a period of rapid change in adapting to the stomachs of langurs. There is a difference of 5 amino acids which are outside the active site of lysozymes of langurs and cows. For making the proteins more resistant to the attack of pancreatic enzymes such as trypsin, several of the arginines were substituted by lysine. Changes in the functioning of proteins were analyzed by understanding the functional significance of amino acid substitutions. Molecular evolutionists can explain the observed changes in amino acid sequences in terms of changes in the functioning of the protein. Evidences based on morphological, physiological, and fossil studies suggest that the two organisms langur and cows do not have a common ancestor, but the lysozymes from these two animals share common amino acid residues that neither animal shares with the lysozymes of their own closer relatives. Despite of having very different ancestry, these two animals have lysozymes converged on a similar sequence. In other words, it can be said that they are homoplaces. The ability of fermentation in the foregut is due to the amino acid residues they share. If one looks at lysozymes in the crop of the birds like a leaf-eating South American cuckoo, an even more remarkable story emerges. These are the only avian foregut fermenters. An enlarged esophageal chamber is present in many birds and is called as crop. The crop of Watsines contains bacteria, thus it can act as a fermenting chamber. Many of the amino acid changes that occurred in the adaptation of Watsine crop lysozyme are identical to the changes that evolved in ruminants and langurs. Thus, despite of independent evolution of Watsine and the foregut fermenting mammals for more than 100 million years, they have each evolved a similar molecule that enables them to recover nutrients from their fermenting bacteria in a highly acidic environment. The example of lysozyme also concludes that inferring single molecules for determining evolutionary origins can be too misleading. Identical molecular changes in this protein are not evidence of common descent but are acquired due to similar physiological conditions. 6.5.3 The evolution of genome size. The sizes of the genomes of different organisms is increased during evolution due to multiple reasons. The numbers of functional genes also increased as the evolution proceeded towards complex organisms. 1. Complex organisms have more DNA than do simpler ones. Among the organisms, genome sizes vary tremendously. The first pattern to be detected was that genome sizes are generally correlated with organisms' complexity. In case of the simplest prokaryote Mycoplasma genitalium, the genome size is only about 470 genes, whereas the genome of Rickettsia prowazekii, the prokaryote that causes typhus, has 634 genes. The most advanced organism on Earth, the human himself, has about 21,000 protein-coding genes. Figure 53 shows the relative sizes of several prokaryotic and eukaryotic genomes. It is not surprising that more complex genetic instructions are needed for building and maintaining a large, complex organism than a small, simple one. There are certain organisms which, being less complex in terms of physiology than the human, have more complex and large genomes than human. 4. Example, lungfishes, some salamanders, and lilies have about 40 times as much DNA as humans do. Figure 53. Complex organisms have more genes than the simpler organisms. The complexity of DNA and the genome sizes vanish when the DNA is transcribed into RNA and the entire genome does not reflect into protein. Only the coding DNA, which makes sense, stands for RNA and ultimately reflects into protein. Eukaryotes have more coding DNA than prokaryotes, plants have more coding DNA than single-celled organisms. Invertebrates with wings, legs, and eyes have more coding DNA than nematodes, and vertebrates have more coding DNA than invertebrates. The organisms with the largest amount of nuclear DNA, some ferns and flowering plants, have 80,000 times as much as the simplest organisms, but no species has more than 20 times as many protein-coding genes as a bacterium. Therefore, most of the variation in genome size lies not in the number of functional genes, but in the amount of non-coding DNA. Figure 54. As the genome size increases, the proportion of non-coding DNA increases. In case of eukaryotic organisms, most of the DNA appears to be non-functional because much of it may consist of pseudogenes that are simply carried in the genome because the cost of doing so is very small. Some of the non-coding DNA may consist of parasitic transposable elements that spread through populations because they reproduce faster than the host genome. To determine the rate at which species lose their DNA, investigators can use one type of transposable element, called as the retrotransposons. By forming intermediated RNA structures, retrotransposons copy themselves and commonly form retrotransposons with duplicated sequences at each end, called long terminal repeats, LTRs. Occasionally, LTRs may join together in the host genome to excise the DNA segment they are flanking. When this happens, one of the LTRs is left behind. Thus, the number of such forfeit LTRs in a genome is a measure of how many retrotransposons have been lost. When the genomes of Hawaiian crickets of the genus Lapala and fruit flies, Drosophila, were compared to study the Number of LTRs, investigators found that Drosophila loses its DNA 40 times faster than Lapala. This rate reflects into the genome sizes, as the genome of Lapala is 11 times larger than that of Drosophila. 2. Genome duplication can be identified on basis of sequence similarity and physical proximity of gene. If the genes are duplicated, the identical copies of the gene have almost same sequence and are physically close to each other. The gene duplication event increases genome size and can have any one of 3 different fates. 1. Either both copies of the gene may retain their original function, with the result that the organism produces larger quantities of the gene's RNA or protein product. 2. One of the copies of the gene may be incapacitated by the accumulation of deleterious mutations and become a functionless pseudogene. 3. One copy of the gene may retain its original function while the second copy becomes severely mutated, eventually acquires some other function and acts as some other protein. This third possibility has highest significance in terms of evolution, as it generates diversity. 3. Rate of gene duplications and possible outcomes. Counting the number of synonymous nucleotide base changes in the genome of an organism can significantly give answers to the question that often gene duplications arise. The number is then compared with the number of base changes that caused protein alterations to see which number changed faster. In case of Drosophila and yeast, the findings were surprising rates of gene duplication which are fast enough to acquire several hundred duplicate genes over the course of 1 million years. In fact, the studies revealed that the duplicated genes were too young, i.e., they are recently being duplicated. Extra genes typically are lost from a genome within 10 million years, which is rapid on an evolutionary timescale. Despite of elimination of extra genes over the years, some new genes were evolved and attained new functions. After many gene duplication followed by mutation events, a group of homologous genes with related functions, which is called as gene family. These gene families are tandemly arranged. Chromosomes, for example, the globin gene family. A homologous gene is a gene inherited in 2 species by a common ancestor. While homologous genes can be similar in sequence, similar sequences are not necessarily homologous. Homologous sequences are orthologous if they were separated by a speciation event. When a species diverges into 2 separate species, the copies of a single gene in the 2 resulting species are said to be orthologous. Figure 55a. Figure 55, homologous genes, A. Orthologous gene, B. Paralogous gene. Orthologous genes are genes in different species that originated by vertical descent from a single gene of the last common ancestor. For example, cytochrome C of human and cytochrome C of horse. Orthologous sequences provide useful information in taxonomic classification and phylogenetic studies of organisms. The pattern of genetic divergence can be used to trace the relatedness of organisms. 2 organisms that are very closely related are likely to display very similar DNA sequences between 2 orthologs. Conversely, an organism that is further removed evolutionarily from another organism is likely to display a greater divergence in the sequence of the orthologs being studied. Homologous genes that are found in the same genome due to gene duplication are called paralogous genes. If a gene in an organism is duplicated to occupy 2 different positions in the same genome, then the 2 copies are paralogous. Paralogous genes often belong to the same species, figure 55b. For example, the hemoglobin gene and the myoglobin gene of human. Paralogs typically have the same or similar function, but sometimes do not, due to lack of the original selective pressure upon one copy of the duplicated gene. This copy is free to mutate and acquire new functions. Paralogous sequences provide useful insight into the way genomes evolve. The globins were among the first proteins to be sequenced and compared. Comparisons of their primary structures suggest that the different globins arose via gene duplication events. Even the timescale of duplication and differentiation events can be revealed by sequencing and studying the amino acid differences. How long the globins have been evolving separately can also be inferred by comparing their amino acid sequences. If more number of amino acids vary in two globin proteins, it means that the two proteins are diverged far earlier in the history. Hemoglobin, which is oxygen carrier protein in blood, is a tetramer that has two aglobin chains and two globin chains. Myoglobin, which is the primary oxygen storage protein in muscles, is a monomer and has higher affinity towards O2 than that of hemoglobin. We can create a gene tree based on the estimated number of base substitutions for estimating the time of duplication of the globin gene, which gave rise to the A and beta globin gene clusters. The rate of amino acid substitution based on the gene tree can be stated as relatively constant, since then about 100 substitutions per 500 million years. The two globin gene clusters are estimated to have split about 450 Maya, figure 56. Figure 56, a globin gene family. Globin gene tree suggests that a globin and B globin cluster diverged about 450 Maya at about the time of the origin of the vertebrates. 6. The uses of molecular information. A variety of biological problems can be solved in various effective ways if information about the genomes of organisms is available. For reconstruction of phylogenetic trees, the information about the rates at which molecules have evolved can be used significantly. Even to determine the phylogenies of genes, the molecular genomic information can also be used. Because genes can be transferred from one evolutionary lineage to another, a phenomenon known as lateral gene transfer. It is supposed on the basis of recent evidences that lateral transfers may have occurred repeatedly during the evolution of plants. The molecular information can be used for 6.6.1 Reconstructing phylogenies. By comparing the structures of molecules from different species, we both gain insights into how those molecules function and acquire a tool for inferring phylogenies. For determining the evolutionary relatedness and reconstructing the phylogeny of two organisms that have been diverged long before, molecules that have evolved slowly can be used. Whereas to study the relatedness of recently evolved organisms, rapidly evolving molecules can be used. Molecules, which are present in all life forms, can be used to study the evolutionary relationships of all existing organisms, for instance ribosomal RNA. Equally important to our choice are strong functional constraints, such as those that exist for rRNA. If even a minute changes occur in rRNA sequence, they may lead to formation of non-functional ribosomes, which are eventually eliminated by natural selection. Since changes in rRNA sequences are allowed slowly, therefore differences among the rRNAs of living organisms can be used to estimate the relative timing of lineage splits that may have happened billions of years ago. There are no fossil evidences which can give idea about the most ancient splits in the lineages of life on Earth into the three major domains, i.e., the bacteria, the archaea, and the eukarya. Therefore we have to depend on molecules such as rRNA for insight into these events in order to reconstruct better phylogenies. Molecular data are used in combination with morphological and fossil data. The more characters we use to reconstruct a phylogeny, the less is the chance of misleading due to evolutionary reversals or convergent evolution. 6.6.2 Determination of the phylogenetic histories of genes. A gene tree shows the evolutionary relationships of members of a gene family, just as a phylogenetic tree depicts the evolutionary relationships of members of a lineage. All of the genes of a particular family have similar sequences because they have a common ancestry. The genes arose from a single gene in their common ancestor but found in different organisms are called orthologs. Genes that are related through gene Duplication events in a single lineage are called paralogs. 6.6.3 provides new ways to combat diseases. We have already seen that knowing the genomes of Anopheles and Plasmodium have medical benefits. Antiplasmodium molecules were produced by creating transgenic mosquitoes, which makes them inefficient vectors of malaria in the laboratory. The emergence of the severe acute respiratory syndrome coronavirus 2, SARS-CoV-2, late December 2019 in Wuhan, China, marked the introduction of a highly pathogenic coronavirus into the human population in the 21st century. The genome of coronaviruses is a non-segmented, single-stranded RNA molecule with positive sense, plus ssRNA, which is, of the same sense as the mRNA. Structurally, it is similar to most eukaryotic mRNAs, in having 5 foot caps and 3 polyadenine tails. The highly pathogenic human SARS-CoV-2 recognized the same receptor, the human angiotensin-converting enzyme 2, AC2, a type I membrane protein found in lung, heart, kidneys, and intestine. A COVID-19 vaccine has been prepared, intended to provide acquired immunity against severe acute respiratory syndrome coronavirus 2, SARS-CoV-2. The vaccine against COVID-19 by referring the information of the genomic sequence of its causative agent SARS-CoV-2. 6.6.4 Molecular data cannot solve all disease problems, although molecular data are of proof themselves effective in solving many problems with several diseases, but when AIDS is concerned, the epidemic suggests that they cannot solve all the medical problems. AIDS is caused by a retrovirus called HIV, which has RNA as its genetic material. The HIV genome constitutes of 2 identical copies of the RNA genome as well as 3 enzymes needed to carry out the viral life cycle: reverse transcriptase, integrase, and a protease. The highly active antiretroviral therapy, HART, is based on the principle to block the viral proteins such as protease inhibitors and reverse transcriptase inhibitors, each of which blocks a different stage in the viral life cycle. But being highly epidemic, the HIV RNA undergoes shift and drifts so that resistant strains of HIV develop in the blood of most patients that receive HART. The mutation rate of HIV is very high, and since it has no repair enzymes, a RON base is inserted at about 1 out of every 8,000 nucleotide positions. Therefore, every time HIV genome replicate a new mutant that is entirely different from the original infecting virus is generated, i.e., no 2 viruses are identical. Thus, HIV's extremely high mutation rate has prevented them from coming up with drugs to which the virus cannot rapidly adapt. Therefore, there isn't any effective medication against HIV. With the advances in sequencing techniques, the genomes of an increasing number of species have been sequenced, and powerful computers have ushered in a new era in the scientific study of the diversity of life. Scientific understanding of the evolutionary patterns of life on Earth and how the agents of evolution have governed these patterns is advancing more rapidly than at any prior time during the study of evolution. Do modern chemical, biochemical, and microscopic methods. We will be able to measure more characters of organisms than we could previously, therefore the amount of molecular data available for systematists is likely go on increasing.